Bismillahirrahmanirrahim. In this video, we will learn about glycosaminoglycans. These are the references for this video. Glycosaminoglycans are also termed as mucopolysaccharides because they were first isolated from the mucus secretion, and they are found in abundant quantity in the mucus. They are long linear polymers of repeated disaccharides. Each disaccharide typically include a uronic acid. They are negatively charged due to carboxylate group and sulfation. The one glycosaminoglycan, that is hyaluronan, is not sulfated. Chemical modification in these glycosaminoglycans are made via sulfation and or O-link sulfation, acetylation, and epimerization. They are highly diverse and specific to the tissue, and there is modification variability and chain composition. When sulfated, glycosaminoglycan chain are O-linked to serine residue of protein cores. Their biological role contributes to extracellular matrix structure, hydration, signaling of the cell, and mechanical resilience. As these molecules are termed as mucopolysaccharides, their retention disease or their accumulation disease within the cell is termed as mucopolysaccharidosis. There are about 12 types of this disease, and they are caused due to deficiency of the enzymes lysosomal hydrolase, and these diseases are inherited mostly as autosomal recessive disorder or maybe as X-linked disease. Clinically, there will be accumulation of the glycosaminoglycan within the lysosome that can be seen in the biopsy, and there will be connective tissue damage, and it readily affects bone, cartilage. It affects stature of the body, and typical faces are found. It may cause psychomotor delays or mild intellectual disability. Life expectancy in these disorders is reduced due to progressive systemic involvement. Hyaluronan or hyaluronic acid is the only glycosaminoglycan that covalently not linked to a protein. There is no covalent binding in this. It is a linear polymer of glucuronic acid and an acetyl glucosamine, and there are about 2,000 to 25,000 disaccharide units in its chain. They are highly negatively charged due to presence of carboxyl group and extremely hydrophilic. There is no covalent protein link. They are synthesized at the plasma membrane, not in the Golgi like other glycosaminoglycans. They are associated with non-covalently bind proteoglycans such as aggrecan, versican, via link proteins or adapter proteins. They are present in cartilage, joint, vitreous humor of the eye, developing tissue, and provisional matrix. The function of hyaluronic acid is hydration to maintain the tissue turgor and resilience, joint lubrication, and cell migration. They are present in abundance during embryogenesis and tissue morphogenesis, hence they have some role in the development of the tissue. They are key component of provisional matrix and facilitate repair and reepithelialization that emphasizes their role in wound healing. Hyaluronic acid may be increased in inflammation, cancer, and scarring, and these are removed by CD44 monocytes. They are degraded by action of enzyme hyaluronidase, and if there is reduced activity of this enzyme, there will be less scarring in the wound. They are used clinically in dermal fillers, viscous supplements that are used for the joints, and vitreous substitutes. They form hydrated gels due to water retention, and they are key in the glycocalyx structure of the cell. Now we will see different types of the glycosaminoglycans and their characteristic. We have studied hyaluronan or hyaluronic acid in detail, that is present in the connective tissue, synovial fluid, and vitreous humor, and it is the only glycosaminoglycan that is not sulfated. Chondroitin sulfate, it contains glucuronic acid and an acetyl galactosamine. It is sulfated and present in cartilage, bone, cornea, skin, and blood vessel. It provides tensile strength and resistance to compression in the cartilage. It is found often attached to the aggregates and other cartilage proteoglycan. Dermatan sulfate contains hydronic acid and N-acetyl galactosamine, and it is also sulfated, present in skin, blood vessel, heart valve, and tendons. Its function is to bind with growth factors and regulate the behavior of the cell. It is structurally similar to the chondroitin sulfate, except this hydronic acid domain. Heparan sulfate contains glucuronic, hydronic acid, and N-acetyl glucosamine. It is highly sulfated, located in the basement membrane and cell surface, regulate signaling of the cell, bind with fibroblast growth factor, modulate angiogenesis and inflammation, and it has got similar structure to heparin but less sulfated, associated with syndecan and glypican. Heparin contains hydronic acid plus sulfated glucosamine. It is highly sulfated, and it is present in mast cell granules. It has got anticoagulant activity, means it inhibits thrombin, and also it regulates immune system. This heparin is most sulfated glycosaminoglycan, and it is used clinically as an anticoagulant. Keratan sulfate contains galactose and N-acetyl glucosamine. This is also sulfated and present in cornea, cartilage, and intervertebral disc, maintain transparency of the cornea, and contribute to the structure, especially in the cartilage. It doesn't contain uronic acid. Least negatively charged sulfated glycosaminoglycan is keratan sulfate. Mostly sulfated is heparin, and least sulfated is keratan sulfate, or non-sulfated is hyaluronic acid. Thank you for watching this video. If you have any question, please write in the comment box.